Good morning, dear students. Today we can study about the biodegradation of xenobiotics by microorganisms. Xenobiotics are chemicals found in the environment which are not produced in organisms. Xenobiotics, the word comes from Greek word xeno means guest, friend or foreigner. Xenobiotics are substances foreign to an entire biological system, that is artificial substances which does not exist in nature before their synthesis by humans. Xenobiotics refers to the unnatural, foreign and synthetic chemicals such as pesticides, herbicides, refrigerants, solvents and other organic compounds. Examples of xenobiotics include pollutants such as dioxins and polychlorinated biphenyls. Xenobiotics are actually man-made compounds which are not a part of the natural environment. They are foreign to the entire biological system and mostly they are pollutant, and sometimes xenobiotics are non-degradable also. So they cause several environmental hazards and problems, and they usually include chemicals such as pesticides, herbicides, refrigerants, solvents and other organic compounds, and example are dioxins, polychlorinated biphenyls, etc. Naturally occurring chemicals can also act as xenobiotics, and these chemicals are called endobiotics. When they are present in excess concentration in the environment, these naturally occurring chemicals can act as a xenobiotic. Natural compounds can also become xenobiotic if they are taken up by another organism. For example, uptake of natural human hormones by fishes. So, naturally occurring compounds can become xenobiotic in two ways. In first way is that if the naturally occurring compound is present in an environment in excess amount, then it can become a xenobiotic. And naturally occurring compound present in one organism can act as a xenobiotic if it enters into the body of another organism. So it just behaves as a foreign material to the second organism. For example, if human hormones happens to enter into the body of any other organism, for example fish, it can act as a xenobiotic. Xenobiotic metabolism. So inside our body, these xenobiotics, they are usually metabolized by the liver. Hepatic enzymes are responsible for the metabolism of the xenobiotics, either by oxidation, reduction, hydrolysis or hydration. The xenobiotics is converted to a secondary metabolite, and then conjugate that active secondary metabolite with compounds like glucuronic acid, then sulfuric acid or glutathione. And such compound, when conjugated with these compound, can be excreted from human body through bile, urine, feces, breath or sweat. So this is the way in which the xenobiotics, if happens to enter in our body, can be removed from our body. But most of the xenobiotics are highly toxic, and detoxification in liver may create problems. So the liver is responsible for the detoxification of xenobiotics. Hepatic enzymes, they are capable of activating the xenobiotics either by oxidation, reduction, hydrolysis or hydration, and convert it into an active secondary metabolite which can be conjugated with compounds like glucuronic acid, sulfuric acid or glutathione, and finally it can be excreted from human body. Biodegradation of xenobiotics. So microorganisms, they are able to biodegrade these xenobiotics, thereby reducing the effect of these xenobiotics in the environment. Microbial degradation of xenobiotics assumes significance since it provides an effective economic means of disposing the toxic chemicals, particularly the environmental pollutants. So by microbial degradation, microorganisms, both bacteria and some fungi, are capable of degrading the xenobiotics, and it is very effective and cost effective means of disposing these toxic chemicals and thereby we can enhance the quality of the environment. So microorganisms are able to degrade this xenobiotics through horizontal gene transfer, by which they make use of such compounds as energy source. So by attaining certain genes from other organisms, microorganisms are able to use these xenobiotics as a source for their energy requirement. So by using these xenobiotics as a source of carbon, they can degrade the xenobiotics, and thus they can be converted into a less toxic material. Microbial degradation of xenobiotics. Microorganisms apply two modes of action for the degradation of xenobiotics, either aerobic degradation or anaerobic degradation. In aerobic degradation, it requires a continuous supply of oxygen, so oxygen delivery systems has to be employed. And in anaerobic biodegradation, here there is no need for oxygen, so an oxygen-free environment is required, and it includes sludge digesters, then groundwater treatment plants, sediments, water-laden soils, and landfills. Here, anaerobic biodegradation is taking place. And some xenobiotic compounds, example tetrachloroethane, polychlorinated biphenyls, etc., can be effectively transformed or mineralized by anaerobic bacteria. So this tetrachloroethane and this polychlorinated biphenyls, they are highly toxic compounds, and they can be effectively converted to less toxic material, or they can be effectively biodegraded by this anaerobic bacteria. So this anaerobic biodegradation is used in sludge digesters, that is in solid waste, as well as sewage wastewater treatment plants, and water-laden soils, that is like marshy lands or wetlands, and also in landfills. We can apply this anaerobic degradation mechanism, whereby xenobiotics can get degraded very easily by the anaerobic bacteria. Xenobiotic degradation microbes. Biodegradation can be done by two ways, either by isolating the naturally occurring xenobiotic degrading microbes, or by the production of genetically engineered microorganisms. So bioremediation of xenobiotics, it can be achieved by either by isolating the naturally occurring microorganism that is available in the soil itself, or by producing such type of genetically modified xenobiotic degrading microbes. So research has been conducted to identify the genes responsible for the biodegradation of the xenobiotics, and microorganism has been further modified by incorporating these genes so as to enhance their metabolic pathway for the degradation of harmful xenobiotics at a more desirable rate. Bacterial species in bioremediation. Members of the genus Pseudomonas are the most predominant microorganism that can degrade xenobiotic. So Pseudomonas, it is a soil bacteria. It is a very commonly available bacteria, and it is very effective in degrading xenobiotic. It is present in almost all soils, and it can degrade xenobiotic. And different strains of Pseudomonas that are capable of detoxifying more than 100 organic compound has been identified. So Pseudomonas putida or Superbug, it is the first GMO. Actually, it is used as a xenobiotic degradant. That is, it is used to remove these oil spills. It is otherwise called oil-eating bacteria. So it is a xenobiotic remediant. Here the xenobiotic is a naturally occurring compound. Oil is a naturally occurring compound, but it is been introduced in excess amount in a marine environment. So in marine environment, the oils are not supposed to present. So as it is present in an excess amount, even though it is a naturally occurring compound, we can treat oil as a xenobiotic. So Pseudomonas putida is the first genetically modified microorganism that is developed as a developed to biodegrade xenobiotics. So, other examples of xenobiotic degrading microorganisms are Mycobacterium, alkali genes and Nocardia. So, more than 100 different organic compounds can be degraded by using the Pseudomonas species, and examples of organic compounds that can be degraded using these bacteria, these different bacteria, that is Pseudomonas, Mycobacterium, alkali genes and Nocardia, include organic compounds such as hydrocarbons, phenols, organophosphate, then polychlorinated biphenyls and polycyclic aromatics and naphthalene. So, a variety of xenobiotics can be degraded with the help of different bacterial species. Next is factors affecting biodegradation. So, there are various factors that influences the biodegradation of xenobiotics. They include the chemical nature of the xenobiotic, which is the most important factor. So, the chemical nature, depending upon the chemical nature of the xenobiotic, the microorganism can either fastly or slowly degrade the xenobiotic. The next is the capacity of individual microorganisms. Then next is nutrient and oxygen supply. Then temperature of the environment, pH of the medium, then redox potential. So, all these factors influence the biodegradation by microorganism. So, my most important feature is the chemical nature of the xenobiotic that is to be degraded. So, factors affecting biodegradation include chemical nature of xenobiotics, then capacity of individual microorganism to degrade these xenobiotics, and the nutrient and oxygen supply, temperature, pH and redox potential. Then xenobiotic degradation. So, water soluble compounds, we have already mentioned that chemical nature of the xenobiotic is an important factor. So, the water, if the xenobiotic is a water soluble compound, it can be easily degraded, and if it is an aliphatic compound, it can be easily degraded compared to an aromatic compound, and presence of ring structures or chains or branches, it will decrease the efficiency of degradation by microbes. So, if the xenobiotic is having a ring structure or a cyclic structure, or it is having long chains, lengthy chains or branches, then the xenobiotic degradation will be much slower. Then molecular orientation of the aromatic compound influences biodegradation. So, ortho compound will be much more biodegraded than para, and least biodegraded compound will be meta. So, this molecular orientation of the organic compounds influences, that is whether this functional group is present in ortho, para and meta position, it will affect the degradation process. So, if a functional group is present in the ortho position, it will be easily degraded when compared to that in para and meta position. So, the presence of certain groups, example halogens in aromatic compound, inhibit biodegradation. So, presence of functional groups also influences the position of the functional group, and presence of the functional group also influence the xenobiotic degradation. If halogen groups are present, then it will inhibit this biodegradation process. Next is Syntrophic bacterial Consortia. Xenobiotics, they may be limited in distribution in the environment. So, degradative microorganisms can be engineered to increase the mobility to access this compound by enhanced chemotaxis. So, if in most of the cases the xenobiotics will be present in the environment in very little amount. So, microorganisms cannot able to detect this compound very easily. So, we have to increase the access of microorganisms to this compound by enhanced chemotaxis. So, by some chemical attraction or attractants, this microorganism should be get attracted towards this particular xenobiotic. So, a particular strain of microorganism may degrade one or more compounds. In some cases, for example, Pseudomonas itself can degrade more than 100 different compounds. So, in some cases, a particular strain of microorganism can degrade more than one compound, and sometimes a single microorganism may not be able to completely degrade a single xenobiotic compound. So, some cases a single xenobiotic compound cannot be degraded by a single strain of microorganism. And in such cases, Syntrophic bacterial consortia must be needed. So, this Syntrophic bacterial consortia means here action with the help of a lot of microorganisms. This degradation of that single xenobiotic compound will be more efficient. So, here by the synergistic action of a group of microorganisms, the biodegradation process is being carried out. That is called Syntrophic bacterial consortia. So, more than one bacteria or one species of bacteria is taking place in the conversion or that degradation process. Here, the end product from one microorganism can be further degraded by another microorganism. So, here the end product by one microorganism is still be a pollutant, but that microorganism cannot degrade it further, but it can be further degraded into simpler compounds or less toxic forms by another group of bacteria. So, the end product from one bacteria can be converted into less toxic compound by another group of bacteria that is living in association with this first group. So, insecticide parathion, it is more efficiently degraded by the combined action of Pseudomonas aeruginosa and Pseudomonas stulceri. So, by the combined action of Pseudomonas aeruginosa and Pseudomonas stulceri, this insecticide parathion can be efficiently degraded. So, it is an example of Syntrophic bacterial consortia for the degradation of a xenobiotic compound. Next is recalcitrant xenobiotics. There are certain compounds that do not degrade, they do not undergo biodegradation, and therefore they persist in the environment for a longer period. Sometimes it may remain in the environment for years without any change, and they are labeled as recalcitrant. So, recalcitrants are bioresistant varieties or biodegradable resistant varieties of xenobiotics which can remain in the environment for longer periods of time without any change, or they can undergo change, but they remain in the environment for a longer period of time without undergoing bioremediation. So, they can undergo changes like oxidation, reduction, condensation like that, but their toxicity will remain like that and they will not undergo any type of biodegradation. So, that type of compounds are called recalcitrant xenobiotics. So, they exist in environment for longer periods without any biodegradation. So, that is all for the today's topic. So, these are the reference tests which I have referred. So, all the notes were taken from different sites by eminent writers, and also notes were taken from Wikipedia. So thank you.